Welcome, everyone. Thanks for joining us here at our science plenary session. My name is Claire Gelfman, and I'm Chief Scientific Officer here at the Foundation Fighting Blindness. And I want to welcome all of you to our session on genetic testing and counseling and my Retina tracker. This session will last approximately 65 minutes, and the last 20 minutes of the session will be reserved for questions from all of you. Please note that this session is being audio recorded, and if you are using an assisted listening device, please turn to channel number 5. And please don't forget to silence your cell phones. The speakers for this session are Dr. Rob Hufnagel and Dr. Todd Durham. I'll give you a brief history of each of their backgrounds, and then we'll get started. Dr. Hufnagel is a 2021 Lasker Scholar, is dedicated to understanding mechanisms of human genome variation that cause blindness in children. He uses a translational approach combining clinical molecular approaches and developmental biology to improve diagnosis and establish gene disease and genotype-phenotype relationships. In particular, he uses genomics, stem cell engineering, and gene editing to establish patient-centered disease models for translational and preclinical studies. After receiving his MD and PhD from the University of Cincinnati, he completed his pediatrics and clinical genetics residency in Cincinnati's Children's Hospital, followed by fellowships in clinical ophthalmic genetics at the National Eye Institute and clinical molecular genetics and genomics at the National Human Genome Research Institute. He loves spending time with his family and their dog named Helix. Dr. Todd Durham is our Senior Vice President here at the Foundation Fighting Blindness. And our foundation, as you know, is a national nonprofit that funds research for the treatments and cures and preventions of both inherited retinal diseases as well as the dry form of age-related macular degeneration. In his current role, Todd is responsible for directing our clinical consortium of retinal experts, managing the disease registry, which we're going to hear a lot about today, developing strategies to enhance product development, partnering with industry, and providing technical input on partnered programs and investment decisions. Todd has over 25 years of drug development experience, and prior to his current position, he contributed to research on numerous marketed products as Director of Biostatistics with IQVIA's Real-World Evidence Solutions, and was a doctoral fellow with Bristol-Myers Squibb, and has worked in various statistical and leadership roles for Novan, Inspire Pharmaceuticals, Quintiles, and as a self-employed consultant. He earned his BSPH and MS in Biostatistics and a PhD in Health Policy and Management from the University of North Carolina Gilling School of Global Public Health. So before we begin, I'd like to encourage you to be social with us. You can follow the Visions conversation on Twitter, Instagram, and Facebook, and share your experiences using the hashtag Visions2022. So without further ado, welcome everyone, and I present to you Dr. Rob Hufnagel.
Hello, everyone, and thanks for your attention, and thanks for being here. I know it's been a very long day and of really important information and really exciting information, and I also know that Todd and I are the thing between us and you all and mingling and dinner. So I really am excited for the opportunity to talk with and to you, and so Todd and I will present our thoughts on clinical testing, genetic counseling, and my Retina tracker, and then we'd love to open it up for questions and engage with all of you. And if you see me throughout the Visions conference today and tomorrow, please stop me. I'll be here through the end of the day tomorrow. And I also want to apologize. Usually as scientists and clinicians, we present with slides and we rely on this information to sort of trigger our brain to say things. So we don't often have the opportunity to talk directly to people in the audience like we really should when we're sharing this kind of important information. So thank you also for this opportunity. So just as disclosures, so as mentioned, I am a federal employee. And I'm at the National Eye Institute, and I do do some work with the Foundation Fighting Blindness on their, which is unpaid, on their scientific advisory board and working with the consortium. And one of the things, also talking about having no prepared slides, this also gave me the opportunity to think about what were the threads that I was able to sort of pick up on from everybody's comments during the day and during the sessions. And it really is about everybody's journeys, and everyone has different types of journeys, whether you're a patient who's learning about your diagnosis or experiencing vision change and or hearing change or other changes, your families, but also your care team. We're on this journey with you, and we all are experiencing these things very differently. And it's really wonderful to see that there are a lot of treatment journeys that are starting now. There's most of the sessions in the morning and afternoon were about the hopes around treatment, and there is also a lot of information about folks' mental health journeys as well, which a lot more attention is being paid to, and I think should continue to be paid to. So that this treatment odyssey has started is really wonderful for so many people. And sometimes that treatment odyssey is for a treatment that doesn't require knowing what the gene is that causes your retinal degeneration. But sometimes it does. And so those folks who are seeking treatment that relies on the gene, there's also a diagnostic odyssey. So that diagnostic odyssey relies on genetic testing to identify the gene, after which you may or may not be eligible for certain clinical trials or may be eligible for certain clinical trials in the future. So the intersection between these two journeys, diagnosis and treatment, is much more real today than even when I was doing my training 10 years ago. So I think that's a really wonderful celebration and a lot of hope, but also still represents a lot of work that we all need to do over the rest of our careers. So I want to introduce then for this to talk more about the diagnostic odyssey. And, that's something that you do with your care team. And your care team involves many people. So that involves your ophthalmologist, your medical geneticist, genetic counselors, clinical laboratory scientists, basic science laboratory sciences, funding agencies like the Eye Institute, like the Foundation Fighting Blindness. We're all working together to try to provide more information and create really patient-centered research. And I want to touch and stress this point very clearly, that genetic counseling is an enormous part of the care. And so if I could please have a show of hands of folks who have had genetic testing within their lifetimes. All right. So if you have had genetic testing and genetic counseling, keep your hands raised. And if you've not had genetic counseling but you had genetic testing, please raise your hands. So you can see it's roughly half, and genetic testing is standard of care for genetic testing, and it's extremely important for several reasons. And not only is it important for your clinical care, but it's also an unmet need. There is a lack of genetic counselors, and this is a master's level graduate program, and there's a board certification, so you'll see MS, CGC after folks' names. And there's simply not enough genetic counselors to provide all the genetic counseling that's needed. And in particular, the subspecialty of ophthalmic genetics that your entire care team made a choice to focus on rare eye diseases, which is the niche of niches. There are still not enough clinical care providers or genetic counselors in this field. But genetic counseling is important for many reasons, but I'll touch on how it interfaces with your genetic testing. So if you've had genetic counseling, or if you haven't, we'll talk more about it. But genetic counseling is in multiple phases during your care visit. So I won't talk about what genetic counseling is in theory or in discipline, but more what it is in terms of one's diagnostic odyssey. So when somebody is going to see the geneticist or the ophthalmic geneticist or the retina specialist, and they get to work with a genetic counselor, this person will provide an intake and talk about your family history and talk about other medical issues that might be informative to the diagnosis. But they'll also talk about what are your expectations for this visit? What is your support structure? What do you do for a living? How has this impacted your life? Do you plan to have children? Are you worried about other people in your families? Who decided, or who asked you to come today? Or are you coming on your own? Would anyone else in your family react to your diagnosis, and how would they react? Because they really want to help prepare you and work with you to really go through that journey of your diagnosis and with your retinal degeneration. And I would also say that they are in that way the cornerstone of the clinical care, and really the one of the number one patient-facing people in ophthalmic genetics care. So they also, in terms of testing, will do pre-test and post-test counseling. So for pre-test counseling, they'll talk about this is the test that we would recommend. This is what the test can offer you. This is the chance that this test will provide an answer. This is the chance that the test will not provide an answer. Here are the types of answers that you might be presented with. You might be presented with an answer that is a gene that is related to your disease. There may be no answer in the test, or like for many people, there's an uncertain answer where genetic variants are found, but it is uncertain as to whether they might be truly associated with disease. And then there's also post-test counseling, where the discussion with the genetic counselor about on the other side, once that test result comes back, what does this test mean? What is this uncertainty? What could we do next? Does this present some availability for clinical trials or natural history studies? Who else should we test in the family? What do you plan to do next for maybe testing parents, or for testing children, or planning for future children? And these are very important conversations that really help to interpret everything within the genetic test. And so genetic testing then, as we move there, is actually a really fascinating and also very modern development. So for most of us in this room, all of the genes on these genetic testing panels, the 350 odd genes that are present on retinal dystrophy panels, or the now 5,000 genes or so that are associated with rare human Mendelian diseases, that is, diseases that are caused by a single gene or changes in a single gene, they've all been found within most of our lifetimes. So for retinal degeneration, this was in the late 80s that the gene that causes gyrate atrophy was found. Also, some of the genes that cause retinitis pigmentosa, the genes that cause color blindness, were all not too long ago. And then the commercialization of genetic testing was also in line with the Human Genome Project. And as sequencing costs came down, it became possible to do genetic testing on a routine basis for patients. And so this started off as sequencing maybe the handful of genetic mutations across a handful of genes at a time, and maybe we're able to find the answer 5% of the time or even less. And then we started to be able to sequence entire genes. And so that was successful for very specific conditions, like, say, X-linked retinoschisis. But for retinitis pigmentosa, we knew that there were tens of genes, and more genes were being found for retinitis pigmentosa all the time. And so then genetic testing transitioned into next-generation sequencing, which is what you hear about these panel sequencing, or exome, or genome. This is all a way of sequencing many genes at the same time to be able to reduce the cost and increase the number of genes that can be looked at. So in the early 2000s, we were sequencing a handful of spots across the genome, and then we were sequencing one gene at a time, and then around 2015 or so, we were sequencing hundreds of genes, and now we're sequencing the entire genome. And I think it's really important to talk about the accessibility of genetic testing for patients. So some of you may remember or may have participated in the iGene program in the early 2000s. And so one of the issues at stake then was that there were many There were many small genetic testing labs, but genetic testing was extremely expensive, and insurance companies were not readily covering this. So the iGene program, as well as other programs for other rare diseases, had started recruiting patients and banking specimens to be able to provide genetic testing that was free of charge and get that back to the patients, and also to help centralize research efforts. And then in 2015, and Todd will talk about the MyRetina Tracker program, the MyRetina Tracker program started providing diagnostic testing for IRDs, but using a much more comprehensive panel approach, which had become very popular and tenable, but only in the mid-2010s or so. And there are other testing programs that are out there for IRDs and for eye diseases and others. But just to touch on these as being very, very important, even if they take extra time to consent patients or they take extra time to receive results, they're still extremely important because they provide more access to genetic testing for patients who may not otherwise, it might not otherwise be available. And over time, insurance companies have followed suit. So even in some places, Medicaid and Medicare are covering genetic testing. And so we hope that as we know genetic testing be standard of care and we want it to be available for all patients, that we hope more and more that insurance companies and Medicaid services will follow suit and more closely follow the development of genetic testing. Now, if we talk about what it is that we're testing. So we have 20,000 genes, and we have about 300, say for IRDs, 350 genes. If we do all eye disease genes, it's about 500 or so. And when we look at everything that we're sequencing, we have sort of a back of the envelope natural variation among us. So again, we have 20,000 genes. If we sequence all of these genes, we have about 100,000 changes between all of us, between you and me. If we did my exome, anyone's exome here, we'd find about 100,000 different changes. And so that means there's about 5 changes per gene. So when we sequence 300 genes or so, we're going to have about 1,500 different genetic changes we're going to find. And then we're trying to pull out the 1 or 2 variants that we think are causing that person's disease. And there's a number of ways to do that, but it is a very stringent process to try to find that one needle in the haystack, if you will, that one variant that we think is pathogenic among all the variation that just makes us a little bit different from each other. And so it is important also then that we have more and more publicly available databases so that laboratories can talk to each other, the same laboratories that worked with iGene and MyRetina Tracker that had have tested lots and lots of patients across the country and across the world that are able to deposit their interpretation of the variants, whether they are pathogenic or whether they are benign or whether they're uncertain and they're looking for more information. And you can deposit those on different websites like ClinVar through the NIH or through LOVD. So laboratories are able to work together that way to provide more information about genetic testing. And so what we see over time is that genetic testing is evolving in a few ways. One is that we're discovering new genes every year. We find new genes for retinal degeneration or for ocular malformations or for syndromes. But also we get more information about the known genes. And as we progress over time and more patients have genetic testing and more of those testing results are deposited in these public databases, then we're able to see a better pattern of which variants are causing disease and which variants just make us slightly different from each other. And so I think that's very important to understand that if you have a variant of uncertain significance on your report, it doesn't mean that it's going to stay uncertain forever. As we understand more and more about the pattern of things, those things that are actually driving eye disease or other diseases, that we'll be able to better understand that uncertainty. And probably a lot of that uncertainty are just benign variants that just make us, again, a little bit different. But we hope that we're able to start seeing that pattern of which are the things that are really the needles. Another area of development in genetic testing is looking outside the genes. So the genes, these 20,000 genes, that's a big number. But this is only 2% of the DNA material that we have. So if you sequence somebody's genome, you're really sequencing 50 times the amount of DNA that we would have just for genes. So if you take the non-gene parts, it's 50-fold more than those genes. So that's an enormous haystack that we have to sift through. So there's now a lot of research trying to understand the pattern in the stuff between the genes. And that's going to evolve over time, and there are many, many laboratories that are working on that now. But I would say that as we've worked our way through the genes, and we'll continue to do that to find new disease genes, we're also circling back to the things that we already know to try to resolve this uncertainty and try to understand what's going on in the things in the DNA in between the genes that are helping to regulate it. And now labs more and more are able to go back and depending on the laboratory, the situation, they may reanalyze reports. Or if they realize the pattern, they may reissue a report based on a change in the way that they interpret the variant. But this is all an area of genetic testing that needs to be more closely standardized, and different labs are working together to really work out those guidelines. And so I think I've covered most of the things that I wanted to cover for genetic testing. But I do want to say that this interface between again, just to go back to the diagnostic odyssey and the treatment odyssey, that this combination is very important for everybody, because we need to be ending the diagnostic odyssey as quickly as possible so that folks can get on with the treatment odyssey. And I think this is all going to be team care and team science with you, with your care team, and with the scientists and the clinical labs that are working with this genes and genetic testing. And I would circle back to the beginning again and say that genetic counseling is as we've talked about in the title of this talk, genetic counseling is a real cornerstone of genetic testing, and it's something that is really, really useful for providing information and for keeping you in the loop about these things. So with that, I will transition over to Todd.
Sorry. That was awful, sorry. Hi, this is Todd Durham. I want to say hello and thank you. If you were expecting and hoping to see Abby Fahim, I'm sorry she couldn't make it. But she's a joy to interact with and a wonderful speaker. I've been at the foundation 3.5 years, and one of the great things about working at the foundation is we get to work with experts like Rob Hufnagel and all the experts in the FFB consortium. It's really been wonderful to learn about this area. So I'd like to share with you today some information about our registry, My Retina Tracker Registry, and our genetic testing program. First, I'd like to tell you a little bit about what a registry is. This is not the thing that you do when you're about to get married and you're hoping to get some china. This has a more scientific purpose than that. So I'll give you a general description of what it is and what our registry is all about and how it works and how we're leveraging our registry to meet our mission. So a registry is an organized system to collect data, so this implies that you have a scientific plan. It is intended to target either a population of people with a certain condition. Some registries target people who are taking a certain therapy. So you may have, say, a newly marketed therapy that generated a safety signal, and the FDA wants you to follow everybody, for example, who took that new medication. Sometimes you use registries to look at pregnancy outcomes as one example. But this really has a scientific purpose. You sort of set these up with these scientific goals in mind, and it's the goals that drive what kind of data you collect. And so you might use a registry to look at a natural history of disease. You might look at quality of care. One of the most well-known registries in the rare disease world has been in operation by the Cystic Fibrosis Foundation, one I worked with many years ago. The CFF developed their registry really to provide information back to their centers of excellence about standards of care, to let them know the centers how they were doing adhering to those treatment recommendations. But a registry can have a lot of purposes. And depending on who's working with the registry, it has different uses and different data demands. So you, say a physician organization was looking at quality of care, they would want a lot of data about what the patient's condition was, what they were treated with, what the outcomes of that treatment were. Sometimes payers like insurance companies and some of the payers in other countries are interested in what was the real outcome of this treatment? Is this worth our continuing to pay for in our health system? So different stakeholders have different needs and different interests in registries, and that drives what kind of data you collect. There are many data types. So from clinical data that would come from, say, a patient's medical record. So that could be directly entered and abstracted from the chart and go directly into this database. You have information that's provided by the patient, him or herself. So they may have symptoms that we want to track over time. And more lately, there's interest in the wearable devices and those sensors that you have in your phones and on your Apple Watches can tell us a little bit about how well we're moving about. And there's some interest in putting those informations in these databases too. But that's just a general description of what registries are, so I'll tell you about what my Retina Tracker registry is. I think you all are experts now on what our mission is. This is really about accelerating the drive to find treatments and cures for the inherited retinal diseases and AMD, dry AMD. And from the clinical side, in both my role with the registry, but also in the Foundation Fighting Blindness Consortium, to me this means removing as many barriers as possible to get therapies into the clinic. And one of the ways we do that through my Retina Tracker registry is to connect people with inherited retinal diseases with research opportunities. And this connection helps remove some of the barriers to address some of the challenges in sort of a disconnected care journey. So some of you may, when you got your initial workup or your first diagnosis, may have seen an inherited retinal disease specialist like Dr. Duncan or somebody at Mass Eye and Ear, but then you return to your community for sort of regular routine follow-up. So we're hoping maybe Dr. Duncan is involved in a clinical trial. Maybe she hasn't seen you in a long time, so you're not on the top of her mind when she's enrolling for a clinical trial. So we're helping to bring that opportunity to you. So the registry was first an online database in 2014. Many years before that, the foundation maintained a contact list of people who wanted to be informed about research opportunities, but 2014 was the first time this became an online database. And the goal then, and as it has been now, is primarily to connect people with inherited retinal diseases with research opportunities. And those research opportunities can be clinical trials, and we know what many people would like. That's what you're hoping for. But sometimes those research opportunities are things like focus groups. A lot of the companies are interested in where do we find you in your care journey to recruit. Some of the companies, some are here represented today, they would like your assistance with designing materials. What would make you interested in a clinical trial? How do we tailor this to make it more accessible and more understandable from your perspective? So these research opportunities can be any number of things. And if you are already signed up in the registry, you probably get regular emails from us asking you to do all kinds of things, and I thank you in advance for saying yes every now and then. I want to emphasize a couple points. The registry is a study. We have IRB oversight. That means you sign an informed consent to participate in this. It also means that we treat your data very seriously. When you share your data with us, your genetic data, your diagnosis, your age, your date of birth, all of that is held confidentially. We have the state-of-the-art health IT infrastructure. We have SOPs that we've developed and all standard operating procedures to make sure our staff don't release information to any entity they should not. When we do link folks in our registry with research opportunities, we don't give them your name. It actually works less directly than that. We let you know. We send you an email to say, here's a research opportunity, and if you're interested, here's how you take the next step. So it's up to you to respond to that or not. The researcher has no idea who you are. We just tell them we contacted 100 people with X-linked RP, for example. But this being a research study, we have an informed consent. That's one of the first things you do when you sign up with the registry. That's the key part of sort of an ethical principle of participating in a human study. And so anytime we change what we collect in the registry, it has to be approved by our institutional review board, mostly to make sure this is not coercive and we're not collecting sensitive information, make sure we describe whatever the risks may be. Our standard operating procedures address all kinds of things about privacy. So our data are in, we have a IT provider whose primary focus is health data, which is one of the reasons why we upgrade our infrastructure back in 2020 to sort of keep up with the times. The new platform we have allows us to integrate the genetics data that comes through our open access genetic testing program that we partner with Blueprint on, and I'll say a bit more about that in a minute. And if some of you have been involved with the foundation for many years, you know one of our longtime employees, Joan Fisher, has supported this program for many, many years, and she's one of my excellent members of my team, and she has really been a wonderful support. So the data that we collect in the registry primarily comes from you. We were interested in your disease journey, what age you were when you were diagnosed, what symptoms did you have when you were diagnosed. We ask things about what assistive devices do you use? Do you drive sometimes now, or are you no longer driving? So any of these things that we can sort of collect about the status of your condition today are really helpful as we think about the natural history, at least in a cross-sectional sense. We do have the capability, and we do have some clinicians who kindly do enter data if you ask them to. And our portal is set up so that your physician or ophthalmologist doesn't need to keep up with your login details. They can actually just go in and type your name and enter in your clinical data. And it's just a handful of information that they're able to enter on your behalf. And our registry coordinators link that up with your profile on the back end. So we're able to know your name and your date of birth, and we link that in. So that's sort of meant to be easy on the clinicians. This program, we're always looking for ways to make this program more valuable to you, to our researchers, and we have a number of new ideas in the works. And one of the great things about the new platform, it allows us to add new modules so it can be flexible. We just completed a study that's been going on, a program that's been going on for 2 years in X-linked retinitis pigmentosa. You may have seen on our website we had a meeting, a webinar last week on June 7th where we got to speak with a lot of folks with XLRP. But before that, we launched a survey through the platform, and the advantage of that, it lets us, we know exactly who has X-linked RP, so we know whoever's receiving this survey is a person of interest rather than someone maybe who had some other form of RP. So my Retina Tracker registry today, it has grown tremendously since it went online in 2014. When we first started the open access and the genetic testing program, no-cost genetic testing program in 2017, we had 4,000 member profiles, and today we have 21,000. And that growth has really been driven by the no-cost genetic testing program. And just to be clear, if you access the genetic testing program, you do not have to register, and vice versa. You can join the registry having completed genetic testing somewhere else. So neither is a requirement for the other. But because this growth has largely been driven because we have attracted, I think, a lot of folks through the genetic testing program. And so I think it described a little bit about how we do link individuals with research opportunities, and how do researchers use this. So some of our partnerships are proprietary in that these are clinical trials, but we have a number of sponsors on our website who have supported the program. But on any given week, we are answering feasibility questions from researchers for various conditions. So, and this keeps me and others on my team quite busy. So I want to say to you, your participation in the registry is really serving our mission. We answer requests probably 4 or 5 per month, and that's a lot of activity for us. And we even take advantage of the registry to recruit for our own sponsored clinical studies like the ProEye study. So if you had EYS-associated RP, we would have informed you this last couple of years that we were sponsoring our own clinical study in EYS and invited you to participate in that. So we use it for our own purposes as well. So I want to describe a little bit more about how this happens. When we have a partner who wants assistance with recruiting, say for a clinical trial as a research opportunity, we require this sponsor to prepare a contact letter, and we have a chance to review that contact letter to make sure it's appropriate, it's written in a way that's understandable. We also require the sponsor to have that letter reviewed and approved by their institutional review board. So we want to make sure you're not being coerced into participating. This approved letter will come from us, usually from a coordinator, is in the from email address, coordinator. What we do is we come to an agreement with the partner on how many people you want to recruit, if there's a particular target. Sometimes it could be geographic, sometimes it's age. If we know something a little bit about the status of your vision, that might be a criterion in which we prioritize our contacts. Again, we don't share with the sponsor or the researcher who you are. We just say, we contacted 100 people with X-linked RP on your behalf, and let us know how it goes. So that's how that works, and then the letter will tell you what next steps you should take if you're interested. Sometimes that next step is to call the sponsor. Sometimes it's to call a contract research organization. One of the language we put in the letter all the time, I'll repeat now, is always speak with your own ophthalmologist about any research opportunity before you change any aspect of your treatment, including a clinical trial. They're going to be your best advisor. So we're just giving this information for informational purposes. So let me shift gears a little bit more to talk about the no-cost genetic testing program, because this is something I think the foundation and supporters of the foundation should really be proud of. This program was started in 2017 as a pilot study at a very small number of centers. There was a real question as to how feasible is it for a nonprofit organization to administer such a program where we're essentially covering the cost of the genetic testing and counseling for those who wouldn't otherwise have access to it. But that initial pilot study was so popular, we expanded the number of physicians who participated until the administrative aspects of the program got so burdensome, we changed this to an open access program that didn't require so many hurdles to jump through to get to the point where you would have access to the test and the counseling. So as I said earlier, if you've received a genetic test and you're not a member of the registry, we have staff who can help you sign up. So it is not a requirement that you go through our genetic testing program to be a part of the registry. But importantly, the data that comes through your genetic testing is an important source of data for research opportunities. So I wanted to, the last couple things I want to say about the impact of this genetic testing program. Thankfully, some researchers at Kellogg at the University of Michigan, Peter Zhao and his colleagues, they looked at the impact of the no-cost genetic testing program on access to tests in their center. So what they did is in the year prior to the genetic testing program and going live in their center, they looked how many people do we have who definitely have inherited retinal diseases and what proportion of them have access to genetic tests. And that number was surprising. Only about a half of their eligible and confirmed IRD patients accessed a genetic test in that year prior to the program. But in the 1 year after, that number had gone up by 29%. So that is just an amazing impact. That this is in a world where some people do have access to genetic tests. Sometimes their insurer will pay for it, and there are other providers and other ways to get a genetic test. But this is an important and very impactful I think, solution to what can be a barrier for a lot of people. And this means there are more people now who know the genetic cause of the disease for their own sake, but it also means now they're potentially eligible for a clinical trial. Now they know what research they're going to be interested in following through the foundation. And so it's super, super important. And last year we asked folks through the registry if they had experience with the genetic testing program, and for many of you who it has, anecdotally people say this really changed my outlook. It gave me an answer where there was just a question mark, like why is this happening to my vision? But one person said it clarified exactly what I had. I thought that I had RP, but found out that I have Usher 2A, so that would be a big clarifying moment. And the other thing is thinking about your family, and I know that's an important issue to a lot of us. One person said, because my family history did not provide a clue about my blindness, the genetic testing has provided relief and understanding for my children and grandchildren, and that's a wonderful gift. So I want to thank you for making this program so valuable to us and to researchers as your participation. I often ask myself, how in the world can the foundation and we keep up with this community of people for years and years and years? And there isn't a secret solution, but I do hope that as a research community, and you're part of it, you stay engaged with us. We have a staff of 3 folks who can help. You can always send an email to coordinator@myretinatracker.org. And I have a wonderful team in Joan, Cindy, and Rebecca Ratchford who are always happy to help out. So I'll end my comments there and happy to take any questions.
Thank you very much, Rob and Todd. Those are great presentations. At this point, we'd really like to hear questions from all of you. We have about 20 minutes. Because we are recording the session, though, please wait till the microphone runner gets to you. But we would love to use this opportunity for you to ask any questions that you might have.
Hi, so you were talking about the registry and putting in when what our diseases and what our symptoms are and things like that. Should we be updating our registry as our disease progresses?
That will be wonderful. The system currently is designed to prompt you to do that once a year. Now we're all partners in this, so I try not to bug people. But if you do occasionally mark it on your calendar, come revisit that once a year, that would be wonderful. The system is designed to track the chronology. So if that changes, all of that's captured. So that would be wonderful.
Okay.
Does the testing have to be ordered by a physician, or can a family just do it themselves?
Great question. The test can be ordered by a healthcare practitioner who's allowed in their state to order a diagnostic test, so not a family member.
Is there any plans to make this just more available for people to do on their own?
Yeah.
I think, generally speaking, direct-to-consumer testing, or DTC testing, is becoming more and more available, such as through 23andMe and others. For specific indications, that hasn't quite expanded to rare disease like IRDs or retinal degenerations. That's a great question, but I think what you're referring to is you would be able to order the test, having seen somebody, and then get that test result back for yourself. And it is a very open area of discussion across all fields of genetics. So there was, at the American College of Medical Genetics meeting this past spring, there were many sessions on direct-to-consumer testing. So it's certainly a conversation that's occurring.
I think we also want to emphasize the importance of having a genetic counselor involved too, because sometimes the results are a little tricky and difficult to understand. So I think that would, from my perspective, would be a concern that one could access the test results but not know what in the world they actually meant.
And Todd was correct in his statement earlier that right now tests are typically ordered through an ordering physician. Often they can also be placed by a genetic counselor working with the physician. And there's also there has been a bill in the works for the possibility of providing genetic counselors with the ability to order testing independently. But right now, I think the general theme that a healthcare provider should be involved with the genetic testing, I think, is important in terms of the expectations for what the test can provide, and also the interpretation. But certainly genetics is creeping more and more into our knowledge, and I'm really always very surprised at what even middle schoolers are learning about DNA these days.
And just to comment on that as well, that if your testing comes from a provider through the foundation, then that testing is free of charge through the open access program that Todd mentioned, and that also comes with the free counseling as well to interpret those results. And then all those results then can go into our registry. So then you've got your information that can help you become accessible for a clinical trial, a natural history study, a research study. So I think it's a great one-stop shop, if you will, to really get a lot of information for yourself and for your family.
I wanted to say thank you so much for providing this information, and kudos to the foundation for offering this genetic testing, and then the counseling, and the tracker is an amazing way to gather data and keep track of. My question is, if one has had genetic testing for a decade or more ago, is the technique or analysis of the current genetic testing any different so that a new genetic testing would be beneficial?
I told Rob he would get this question.
That is a wonderful question, and it is a bit of it depends on the circumstances. So there are some variants that have been disease-causing or known to be disease-causing for decades. Other variants were initially thought to be disease-causing, and then they were found out to be benign. And one of the reasons for that was that more and more sequencing of folks across the world has sort of let us understand what are those variants that just make us a little bit different but don't actually cause disease. And so those databases that have the frequency of these variants in the general population didn't really appear until about 2010 to 2015. So testing prior to that, we did note that some of that did change as far as whether it was the cause or not the cause. But it wasn't for a large number of variants. So I would say, if your genetic test report did say that, these are disease-causing variants and it matches the expected inheritance pattern for that gene, whether you need 2 genetic changes in the gene to cause disease or 1 genetic change to cause disease, then you may not need to have it reanalyzed. It may be valuable if you see a genetic specialist that if they've reviewed it recently, just to say, oh yes, this is still the answer for you. So it doesn't necessarily need to go back to the clinical laboratory. Your provider, genetic counselor, or physician should be able to, especially if they have a physician trained in genetics, that they should be able to interpret that for you. That's an awesome question. It's something we've definitely seen change, especially over 10 years. And did I answer all of that? Sorry. Okay, yeah.
Let's say that a woman, as an example, gets the free genetic test and the results are that she is a carrier of a recessive form, but she does not show any symptoms. And let's further suppose she has 2 daughters, and both of them are interested in knowing if they're carriers. What are the financial implications for, say, the 2 daughters who now want to get tested? In other words, what would it cost them to get the test?
Yeah, so thanks for the question, Dan. Through the current program, Blueprint Genetics offers family variant testing. It's quite a low price, but it's not offered at no cost through the foundation. So I think it's, what did you find out? $150. So there it's very limited because you actually know what you're looking for. So you wouldn't need to do a full clinical panel for such a case.
And since not everyone has had their testing through Blueprint Genetics, other laboratories have different policies, but certainly contacting the laboratory. But I completely agree with Todd that starting with just that variant and not doing the entire test is the best way to go for that particular question.
Okay. I was tested when I was 14 back in 1958, and at the time, no one in my family knew of anybody else who had this until I had kids, and my oldest two kids both have RP. And then in later years, I found out another first cousin had it, and another brother was diagnosed with it. But is it possible that some of our genes can be different in the same family? They mutated and did something different.
So typically when we see a disease like retinitis pigmentosa or something that's in multiple family members, then we do make the at least initial assumption, or at least say that the most likely scenario is that it's the same genetic change in all of the individuals that's leading to that particular disease. There are exceptions to every assumption or to every, I wouldn't call it a rule, but there are certainly cases where there can be multiple causes of retinal degeneration, two different genes causing the same or similar retinal disease that can be present in families. That does happen.
Should I encourage the other in my family to get their genes tested or not? Would be necessary?
I think as more and more there are clinical trials and natural history studies becoming available, and sometimes the eligibility for those trials depends on confirmation of a genetic change in a certain gene, that I do think there is value in them having their own diagnostic report, but they can start with the genetic change that is known to be in the family, or if it is unknown for others, that one person can start with more of a broad sequencing panel, find the answer, or the genetic cause rather, and then other people in the family can have genetic testing for the same change to see if that change goes with those individuals that have the condition. Does that answer your question?
Yes.
Okay, thanks.
Rob, I want to ask you a question.
No.
So I got an email from somebody. They said, we've had whole exome sequencing and we had a commercial panel and we still haven't found the disease-causing gene. And they wanted to know, what's the difference between a whole exome panel or sequencing and a commercial panel, and why would any of us pursue one over the other?
That's a great question. So a lot of, and it's an ongoing debate. So depending on the number of genes that are associated with a certain condition, it becomes more cost effective to simply do exome sequencing, sequence all 20,000 genes, if it's the same cost of sequencing, say 300 genes. So I could maybe call it the secret sauce behind a lot of the genetic testing panels these days at different clinical laboratories is, they're actually doing the sequencing as exome sequencing, but the panel is just the analysis of those 351 genes or 500 genes or 700 genes or 200 genes, depending on the different disease. So now it's becoming more and more commonplace that it's exome sequencing with a panel analysis. And then for certain conditions, so the American College of Medical Genetics has started putting out guidelines for recommendations for testing for different diseases. So for intellectual disability and congenital malformation, so children that are born with birth defects that also have developmental delay or intellectual disability, the first-line genetic test is now thought to be exome sequencing or genome sequencing, and analyzing the entire thing rather than even focusing on a particular panel. So whether we will see that for IRDs or not is a question that a lot of us are asking. And so, we may in a few years even transition to exome sequencing as first line for IRDs, or we may keep it as, let's focus on the 300, 400 known genes that are associated.
We have time for about 2 more questions.
I had a question here. So first, thank you for everything that you and your colleagues are doing to fight blindness. So me and my siblings are here for my mother, and she did a genetic testing, but they couldn't identify the specific gene. And so they wanted her to kind of get the tested from her other siblings, so like her aunts and uncles, but it's difficult for family members to kind of do that if they don't want to. are there other options or courses that we can do just so we can focus on our mother and try to get that genetic identified?
You can start from there right now. What would you do for?
Is it an issue of, practically speaking, it's hard to get a sample from the family members? Is that the issue?
That is one,
Okay.
and so is there any other options out there that can we use our children, or is there?
Yeah, I think starting with individuals who are affected, and typically genetic testing for finding the condition is often done on individuals who are affected, and if there's things like carrier testing or for people who do not have the disease, often the recommendation is to wait until they're adults. So there are differences between some of the genetic testing panels. So to a certain extent, it depends on when the genetic testing was done and which laboratory had done the testing. And we sort of touched on two things. There's I guess, two concepts here. We've talked about reanalysis, but then there's the idea of doing another test. So after enough time, the number of genes that have been are sequenced, or the number of genes that are analyzed, increases, and so the diagnostic yield, or the chance of finding the answer, does slowly increase over time as the technology changes. But I think one of the other reasons why a lot of these panels are sort of changing into an exome sequencing with a panel analysis is that you could go back and reanalyze a larger number of genes instead of repeating the sequencing. So it sort of depends. But if somebody had the Sanger sequencing back in 2010 and did not get an answer then, I think it'd be very appropriate for them to get a much larger next generation sequencing panel or exome type test now.
Thank you.
Sure.
Question? Last question? Yes.
My son was diagnosed with Usher's 2, and he has been tested, and no gene has been identified specifically for him. When you do the testing for Usher syndrome, does it include all of the known RP genes also, or just specifically Usher genes?
So it depends on the test. So there are dedicated Usher syndrome panels, and there are Usher syndrome genes on the larger retinal dystrophy panels. And there's, this is often discussed, that one could have a retinal degeneration due to one gene and hearing loss due to a second gene. And so sometimes those hearing loss genes are actually included in the Usher panel or in the retinal dystrophy panel. So it's a fantastic question, and I think that possibility might still exist, that there could be two genes that are affecting hearing and vision. And there are still genes that, and still trying to understand the DNA around the genes for the Usher syndrome genes, and that the answer might be there as well. But as the technology develops, or having broader sequencing testing might help to get a better sense of that.
So time to test again, I think. Okay, thank you.
Sure.
Well, it's time that we conclude our session. I want to thank everyone for being here. I do have one quick announcement. There was a purse that was turned in. If Susan Robinson is in the audience, your purse can be found at the registration desk. So thank you. I think I saw a hand go up there. Well, please join me before we make our way to the welcome reception, which is in the exhibit hall. Please join me in thanking our great speakers, Rob and Todd.